Another type of blood grouping is called RH grouping. RH grouping means RBC can be surrounded by RH antigens. They can be surrounded by RH antigens. Apart from ABO blood grouping system, now RH antigens are made up of proteins, and it's called as RH because it was found in an ape, which is RS rhesus monkey. So that's why it's called as RH antigens. Now those who have this kind of antigens, they are called as RH positive. So suppose if an A blood group person is having these RH antigens, then they will be called as A positive. B blood group person is having, they will be called as B positive, likewise. And those who do not have these RH antigens, if the RBCs are not surrounded by these RH antigens, then they will be called as RH negative people or RH negative blood group. So during the blood transfusion, RH factors also should be taken into consideration, because suppose if the blood of an RH positive person is getting donated to an RH negative person, this is RH positive, and if suppose if it gets donated to an RH negative person, what happens is that RH negative person will be making antibodies against the RH antigens. So they will have antibodies against the RH factors. So these antibodies will clump. They will start clumping with the RH antigens of the donated blood, and when they clump together, they will lead to anemia, and it can be fatal, can lead to the death of the person. So that's why during the blood transfusion, the RH factors also should be taken into consideration. They should be matched. Now there is one case of RH incompatibility. RH incompatibility means mismatch. This happens during a pregnancy period. That is, if the mother happens to be RH negative, if the mother happens to be RH negative and the fetus or the baby yet to be born, if the fetus is RH positive. So what happens in this case is that during the first pregnancy, there won't be a problem, because there won't be mixing up of blood between the mother's placenta, no mixing between mother's blood as well as the fetus blood. But during the delivery stages, so when the delivery stage happens, these placenta, now this is the placenta is supplied with mother's blood, and it is connected to the baby. So if this is the first pregnancy, then the mother's blood, even if the mother's blood is RH negative and if the baby's blood is RH positive, since there is no mixing up of blood between both, there won't be any problem. But during the delivery stages, these blood vessels can rupture. At that time, the mother's blood will be able to recognize that there is some RH positive factors present in the blood. and then as a reaction to that, Rh negative blood of mother in the plasma starts to create antibodies against the Rh factors. So there will be antibodies against the Rh factors created in the mother's blood. But by the time during the first pregnancy, the baby will be delivered out. So there won't be any problem happening to the first fetus. But during the second pregnancy, now that the mother's blood is already prepared with antibodies against Rh factors, these antibodies in the second or subsequent pregnancies, these antibodies can cross the placenta and affect the babies. That is, if the even in the second pregnancy also if the baby is Rh positive, in that case, now that the mother's blood has already prepared antibodies against the Rh factors, these antibodies can cross and come towards the baby's blood. This will result in clumping together of the baby's RBC or the fetus RBC and the antibodies of the mother. The antibodies of the mother will clump onto the RBCs of the fetus. This is fatal for the second fetus, that is for the baby. It will result in anemia and jaundice for the baby. And since it's fatal, this problem is called as erythroblastosis fetalis. Erythroblastosis means erythrocyte is getting destroyed and leading to the death of the fetus, and this is destroyed due to the Rh incompatibility. So that's about the Rh incompatibility during the pregnancy. It happens when the mother becomes Rh negative and the fetus will be Rh positive. Now, in order to prevent such further occurrence, after the first delivery, immediately after first delivery, in order to prevent the clumping together, anti Rh antibodies. Anti-Rh antibodies means they are against the antibodies created by the mother. So anti-Rh antibodies can be injected. Anti-Rh antibodies can be injected to mother immediately after the first delivery. Immediately after first delivery. So in this way, we can prevent the antibodies of mother affecting the antigens of the second baby. So the antibodies of the mother will not be destroying the RBCs of the second fetus. So this is a preventive measure taken in order to prevent the erythroblastosis fetalis.